Stem cells, when they were derived, particular human stem cells, when they were derived, they actually were derived with very similar protocol compared to mouse. In order to maintain their pluripotency, they require a fibroblast, mouse embryonic fibroblast that secrete cytokines to, in order to support the maintenance of pluripotency maintenance in the mouse stem cells as well as in the human stem cells. But when we checked, as particular when we maintain these cells, what particular growth factors needed for mouse stem cells not necessarily the same and what they need for maintaining human stem cells. And there was this biggest issue where they come from. So they do come from inner cell mass of the embryo, but is it the step forward in the human that they were derived from, probably from epiblast of the human embryo, versus in the mouse they were derived from totipotent cells from inner cell mass. And the biggest challenge in order to test this in vitro, that we cannot put human embryonic stem cells back to a human embryo and see if we can see the contribution to chimera, which we can do it in the mouse. And even in the mouse, there is a term of totipotency. So it means that when you actually introduce to mouse blastocysts, not just the blastocysts, you can actually fuse two-cell mouse embryo. This technology was developed by Andras Nagy in 90s when they made the tetraploid embryos, and then it's known that tetraploid embryo actually would participate in the tetraploid cells in the embryo would participate into development of placenta, and then during the blastocyst stage they would actually inject mouse ear cells, and the embryo would actually develop only from the mouse embryonic stem cells that actually been injected. And that way this technology allowed to test mouse ear cells for their totipotency. So we can't even talk about this test in human. We can't even make chimeras in human. So this biggest question in the field still exists. If human ear cells actually, we know that they are pluripotent, so they can make different type of tissue and they can differentiate to pretty much any type of tissue in the body. But can they actually be totipotent? So the mouse stem cells require, in order to maintain their pluripotency, they actually, the pluripotency driven by STAT3, we know that it's a leaf STAT3 pathway. So human stem cells compared to mouse, they actually drive their pluripotency using a nodal activin pathway. So it's a slightly different, so it's a step forward in development. So we think that, and it's been a number of publications in 2010 that confirmed this, that human stem cells probably are, they call them, the stage is a primed ear cells, versus group of Austin Smith has actually shown in a mouse a ground state of pluripotency, that it's they were derived in a bit earlier stage of development, still from blastocysts. And this is also, we can go back to human embryo, and we can, from donated embryos, we can actually try to derive human ear cells that will be actually, we can catch that transient state of development in human, because they have to go through that state as well. So we want to try to catch that stage of the embryo and derive human stem cells at that particular stage of development, ground state pluripotency. And this is challenging because we have to find in vitro system first that actually, and need to know what growth factors and what, not just the growth factor in the media, but also the substrate that they need in order to maintain this ground state of pluripotency. There is a difference between human and mouse ear cells, how they behave in vitro. So the human ear cells have very poor cloning efficiency. So what does that mean? So the mouse ear cells can literally grow from a single cell. So for example, we call this method passaging. For example, if you make the stem cells, you want to multiply them. So you use a specific disaggregating enzyme and make them single, and then you actually plate them down. So the human stem cells, you literally cannot do it, or at least you can't do it for a long time. So you will introduce some genetic abnormalities and so on and so on. So, and that is the probably biggest challenge. So you can get human ear cells in a timing from the single cells. You can't make billions of cells in a few, for example, weeks or so, in order to use it for certain protocols to differentiate or for your research. It's one of the biggest issues probably with human stem cells. Another difference is, as I said, they use a different signaling pathway, different growth factor conditions. So all of that, we, not only our group, but also the researchers in stem cell field are working on these issues in order to resolve. So this all will actually benefit in the future IPS technology, disease modeling, and if the human stem cells will be very close to mouse, and I think we will get to that point eventually. The main probably technical issue, or let's say, in creating human stem cells, it might be a biological difference, we don't know. So the human stem cells grow much slower, and they can't grow from the single cell readily as the mouse stem cells do. So they have very low cloning efficiency, and also they require different growth factor conditions in order to maintain them. So mouse ear cells grow much easily. They differentiate much easily. You can differentiate to certain lineages. The protocol work much faster, and I would say much easily. So with the human stem cells, we have to spend quite a bit more time to do this, but we do this anyway. So the biggest, probably our goal will be to find the condition that would be mimicking mouse ear cell condition, and the human stem cells will behave very similarly to mouse, and not just by the growth condition, but also the epigenetic state, as well as their transcription factor, everything, what they express. And this probably would help us if we do it we will find it, these conditions, we can go back to human embryos and derive, when we know exact conditions, we will derive those naive, we call them naive human stem cells in the future. So this will actually help us to understand the human development in general, and maybe human pre-implantation development. Why we need human stem cells condition to be very similar to mouse ear cell condition is one of the probably biggest advantage of this would be, so we can actually manipulate human genome easily. So it means that we can introduce the genes or mutations that actually would be mimicking human diseases. So, and with much more efficient way. And that's probably one of the biggest advantage why we have to address these questions in order for development of the human to diseases is for sure. And this would also apply for iPS technology. So when we know that we have a ground state pluripotency in human ES cells, and we know the conditions that would maintain that ground state pluripotency, that would make much more easy to address lots of biological questions. So it might be a technical challenges, but it also has a big impact for research. So, and then we can go back and actually derive or reprogram induced pluripotent stem cells, and will maintain them in that ground state of pluripotency in order to use those iPS cells for future research on modeling diseases and so on. And I think this is quite important for pretty much every step of development of stem cell therapies.